Welcome, aspirants, to the high-yield explainer for biological classification. If you're looking to lock down your marks and really master the absolute essentials from the NCERT syllabus, you are right where you need to be. Today, we're extracting the most critical concepts, the key structures, and those classic exam traps directly from the source. Let's dive right in.
So, did you know that in all the vast, messy complexity of life on Earth, there's a tiny, single-celled organism that holds enough unique traits to command a kingdom entirely of its own? Seriously, it doesn't quite fit with plants, it isn't an animal, and it's absolutely not a fungus. This beautiful biological messy middle is exactly why a structured classification system had to be invented in the first place.
Here is our high-yield roadmap for today. We are covering Whitaker's master plan, the extreme survival tactics of kingdom Monera, the eukaryotic misfits of Protista, and the diverse decomposers in kingdom Fungi. Then we'll hit the exceptions, viruses and viroids, before wrapping up with a rapid, NEET revision recap.
Let's start with Section 1, Whitaker's master plan, the five-kingdom classification.
You really need to have Whitaker's criteria memorized straight from NCERT. Before his macro rules dropped in 1969, organisms were just awkwardly shoved into two boxes, plants and animals. But Whitaker completely changed the game. He used cell structure, body organization, mode of nutrition, reproduction, and evolutionary relationships to build five distinct pillars. For example, if you think about cell walls, fungi are now separated from plants because fungal cell walls contain chitin, while plant walls have cellulose. And you'll notice Monera is totally isolated as the only kingdom with a prokaryotic cell type, meaning they completely lack a nuclear membrane.
Which brings us to Section 2, Kingdom Monera, Extreme Survival.
This is the domain where bacteria absolutely reign supreme. Now, bacteria might be simple in structure, but their behavior is incredibly complex, and they show the most extensive metabolic diversity of any group of organisms. You've got to recognize their four fundamental shapes. There's the spherical coccus, the rod-shaped bacillus, the spiral spirillum, and the comma-shaped vibrio.
Listen, this next part is a classic trap for students, so don't fall for it. You absolutely have to contrast Archaebacteria with Eubacteria. Archaebacteria are basically extreme survivors. They thrive in hypersalty areas as halophiles, they live in boiling hot springs as thermoacidophiles, and in marshy swamps as methanogens. Their secret weapon is a totally different cell wall structure. On the flip side, we have Eubacteria. These are the true bacteria, and they're characterized by a rigid cell wall and, if they happen to move around, a flagellar.
Within those eubacteria, we find the Cyanobacteria, also known as blue-green algae. A great example of a photosynthetic autotroph in this group is nostoc. The main thing you need to remember about nostoc is its specialized cells called heterocysts. These are incredibly important because they're designed specifically to fix atmospheric nitrogen. So, whenever you see nostoc or anabina on an exam, immediately think nitrogen fixation and heterocysts.
Before we move out of Monera, we can't forget Mycoplasma. These are the pathogenic rule breakers of the bacterial world. They completely lack a cell wall, which makes them super unique here. And they actually hold the title of the smallest living cells known to science that can survive completely without oxygen. Definitely lock that fact in.
Moving on to Section 3, Kingdom Protista, Eukaryotic Misfits.
We've conquered the prokaryotes, so now we're looking at Protista. This kingdom is fascinating because its boundaries are actually pretty blurry. It forms this vital link connecting plants, animals, and fungi. We're mostly talking about aquatic, single-celled eukaryotes here. And because they're eukaryotes, they have a well-defined nucleus and membrane-bound organelles. They really run the gamut, ranging from photosynthetic producers to aggressive predators and parasites.
You really want to pay close attention to the distinctions between the groups here. First up, Chrysippus, which includes diatoms. Their cell walls are actually embedded with silica, making them virtually indestructible. Over billions of years, these shells pile up to form diatomaceous earth. Next, we have dinoflagellates. These guys have stiff cellulose plates on their surface, and rapid multiplication of red dinoflagellates, like Gonyualix, is what causes toxic red tides that wipe out marine life. Finally, euglenoids. They are super interesting because they completely lack a cell wall. Instead, they have this protein-rich layer called a pellicle that makes their bodies highly flexible.
All right, Section 4, Kingdom Fungi, Diverse Decomposers.
Fungi make up a totally unique kingdom of heterotrophic organisms, and they are essentially the ultimate terrestrial recyclers. Whether we're talking about the mold ruining your bread, the yeast brewing a beverage, or the agaricus mushroom sitting right on your dinner plate, fungi show just incredible morphological diversity. They're cosmopolitan, meaning they're found literally everywhere, from the soil and air to the inside of living animals.
So what exactly unifies all these different fungi? Well, keep the word chitin at the top of your mind. Unlike plant cell walls made of cellulose, fungal cell walls are strictly composed of chitin and polysaccharides. Their bodies are made of these thread-like structures called hyphae, and you need to watch out for the term coincidentic hyphae. While normal hyphae have cross walls, coincidentic hyphae don't have them at all. They are literally just long, continuous tubes filled with multinucleated cytoplasm.
When it comes to sexual reproduction in fungi, the process is highly structured and it gets tested all the time. It happens in 3 very exact steps. Step 1 is plasmogamy. This is the fusion of just the protoplasms between 2 gametes. Step 2 is karyogamy, which is the actual fusion of the 2 nuclei. And finally, step 3 is meiosis, a reduction division in the zygote that results in haploid spores.
Now, pay close attention to a very specific delay that happens in some fungi because it's key NCERT terminology. It's called the dikaryophase. And in ascomycetes and basidiomycetes, right after plasmogamy, the nuclei don't fuse immediately. The cell takes a pause in an intervening stage where it contains 2 distinct nuclei per cell. We call this the N+N condition, or the dikaryon. They will eventually fuse, but this temporary phase is a major defining characteristic that you've absolutely got to know.
Next up is Section 5, the exceptions, viruses and viroids.
Here we leave the established kingdoms behind entirely. These biological entities didn't even make the cut for Whitaker's 5-kingdom system. Distinguishing between these two is basically guaranteed exam marks. Viruses are obligate parasites. Outside of a host, they are completely inert crystals. They consist of a protein coat surrounding some genetic material. But crucially, that genetic material can be DNA or RNA, but never both at the same time. Viroids, on the other hand, are even smaller than viruses. They were discovered as infectious agents causing diseases in plants. The vital difference to remember? Viroids are just free RNA, and they strictly lack that protein coat that viruses have.
All right, time for Section 6, Rapid NEET Revision Recap.
Let's hit this checklist fast. Burn these facts into your memory because examiners absolutely love them. Methanogens produce biogas. Nostoc relies on those specialized heterocysts to fix nitrogen. Mycoplasma breaks the rules by lacking a cell wall entirely and being the smallest living cell. Diatoms are armored with indestructible silica, and euglenoids swap out a rigid cell wall for a flexible protein-rich pellicle.
Continuing our sprint through the high-yield facts, fungal cell walls mean chitin, not cellulose. Plasmogamy is purely the fusion of protoplasms, whereas karyogamy is the nuclear fusion. Deuteromycetes are famously called the imperfect fungi because only their asexual or vegetative phases are currently known to us. Remember, no virus on Earth contains both RNA and DNA simultaneously. And finally, viroids are just free RNA. They completely lack a protein coat.
You've made it to the end of our high-yield blueprint, but before you close your notes, I want to leave you with a fundamental paradox of biology. As early scientists proved, viruses can be crystallized and sit perfectly inert on a desk, completely lifeless. Yet the second they breach a host cell, they hijack the machinery, replicate relentlessly, and kill. So based on everything we've decoded in this explainer today, I have to ask you, would you classify viruses as living or non-living? Think about that until your next revision session. Stay focused and keep learning.